Welcome everybody to 2.5 enzymes. Hope you enjoyed 2.4, 2.1 through 2.4. But we need to talk about enzymes. Enzymes are so important to how your body works. And you've learned this before ninth grade, but we gotta keep going after it. So one thing you'll have to always know is the word catalyst. Okay, it means it speeds up a reaction without being used in the reaction. And it speeds it up by lowering the amount of energy. So lowering the amount of energy to start the reaction. And it is not used up, it is recycled in the reaction. And we'll get to some nice diagrams later, but something you'll have to, the vocabulary you'll need. Vocabulary active site. That's the spot where the enzyme, this is the enzyme, matches with the substrate, which is I'm drawing the substrate right here. Believe it or not, this fits into this area here, so that area is the active site. Anyway, the diagrams I'll show you next will be a lot better. And the substrate is what works in joining up into the enzyme. This totally messed up diagram that I made. And the substrate pops into here, reactions occur, and that turns into products. And this is a diagram to show you very simply how the energy works with the enzyme. So here we have our reactants on the left side. So you got reactants, whatever it is, we're starting out with. We have this energy diagram. One, you see the energy go way up, and then another with the energy that goes down. Guess which one has an enzyme, the top one or the bottom one? I told you earlier, enzymes do what? They lower the activation energy. So this is with enzymes here, the blue, and up here is without enzymes. Well, I'll say without an enzyme. And then at the very end, after you have your reactants, this signifies the reaction, you have your products. Reactants and then products. A little bit of activation energy with an enzyme, this much. You need this much without an enzyme. And so 3 stages. The substrate, as I mentioned before, the substrate binds to the active site and it is changed, converted into other substances called those products. And then after that happens, it's separated. And those are the 3 steps. Something to also know is there's always, particles in a liquid or gas are always moving around in motion. When they stop moving, then you have a solid. But they move at random. They just kind of bang into each other. And if a substrate happens to come in contact with an enzyme, goes into the active site, a reaction occurs. And if you heat that mixture, it speeds it up. I can't tell what's behind the box here because the video's blocking it. Here we go. So here we have, it says ligand, but this would be our substrate. Moving into the active site. This is called the locking key. I have a key on me right now, but key goes in the lock, has a certain shape. It works moving the pins around, makes the barrel spin, and then it opens. You mess up that key, you're going to mess it up. Here's another diagram here, substrates. Right here, moving into the active site. And it changes shape sometimes as substrate enters the enzyme. And here you see they've changed. They've changed from a substrate into the products. That's what I have here. So you had the original reactant. Now it is a product, something totally different. The enzyme is the same. It has not changed. Here's what it'll be like in your body. Your body is mostly water because that's the best way for the reactions to occur. And all these little round symbols right here, these are water molecules. And these little things floating around like little boats, look, they're the same shape as this thing right here. This is the substrate. And right here is the active site. And this isn't labeled, this yellow-green thing, but this would be the enzyme. If I did have you label it, which I'm having you label it, you put that in your notes. Normally you'd have the slide packet. Not anymore though. All right, this is something that you are with your, I'm going to sign you up in groups. You're going to be designing an experiment. You're going to look at how, let me find, where am I at here? How temperature, pH, and the concentration of the substrate affect the reaction rate of these enzymes. So when they're heated, all the molecules move faster and more likely to come into contact with each other. And if you have more contact, you're going to have more reactions. So they react at a higher rate. More likely to come into contact, they are going to react. But also too much heat can be bad because they can break by breaking the second. So I think what I meant to say here, there's bonds that are in the, holding that protein together, and that when you break the bonds in the enzymes, it can change the shape, which is called denaturing or denaturation. I have to review that. Maybe I have something else, but you're basically heating it up. You are destroying the shape of that protein, and then it's not going to fit in the way it should. pH works the same way. That high or that low pH or high can alter bonds or bonding and lead to denaturation. Sorry. The concentration of the substrate can also alter the rate. So if you have more if you have more substrate, more things like we saw in the boats earlier floating around in there, that's going to become problematic. And more substance, there'll be a greater chance that they'll enter the active site and then therefore react. Also, if the enzyme concentration is higher, you're going to have more interactions too, increase. So if you have more substrate or more enzymes, you're going to have a higher reaction rate. But if you have all of your active sites filled up with a substrate, you can't have any more reaction. So as the action increases, and that slows down the reaction, but it doesn't like make it decrease. It basically it'll increase, and if you occupy those active sites, that rate will stay steady, and a plateau is reached. EAU, plateau. Here are some examples right here. You look at the pH. Different types of enzymes are made to work in different areas. So this is pepsin. It's found in your stomach, and you'll see it likes about 1.5, which is very acidic. And salivary amylase, that's in your mouth. You don't want stomach. You don't want something that acidic in your stomach. You want a more neutral pH, slightly acidic to help knock down some bacteria, so that your amylase can work well. And this is enzyme that's in your small intestine, which is slightly alkaline. So it's made to work at that pH. And here we have, as the substrate concentration increases, we see increase in reaction rate and then a leveling off. Okay, if you increase it 4 times, you have that much faster. If you double it basically 1 time, it gets a little bit faster. So the more substrate, the faster, but eventually it all levels off because those positions, those active sites, are all filled. I'm going to write that. Active sites all filled. And here's another little diagram. Temperature increases as it increases enzyme activity. So this is, you're basically, is this true? Now remember, enzyme activity will increase to a certain point with temperature, and then as it starts to denature, will decrease. So this is incorrect. This is your correct answer. This is correct here, but it should start to dip. And you have a more of a steady increase, and then you have a drop off. Here it's just showing you get super hot, it levels off. No, it doesn't. It will start to decrease because the enzyme denatures. And on to part 2.